All right, so this is chapter 22 on the lymphatic system. And so here you can see the entire lymphatic system. There's a lot of vessels, and those vessels are called lymphatic vessels. And wherever you have arteries, whatever tissues have blood supply to them, they're going to have lymphatic vessels. Usually the arteries and veins run right alongside the lymphatic vessels. They're a lot like veins are. They're thinner walled. They have valves in them so that fluid won't back up, but the pressure inside them is very low. And then there's a couple of other differences between the lymphatic system and the circulatory system. First of all, in the lymphatic system, there's no red blood cells. There's the white blood cells, the lymphocytes. Those are the main cells that we find inside those lymphatic vessels. And then there's a fluid in there that's a lot like interstitial fluid. Because the interstitial fluid, it's a connective tissue. It flows. So the plasma from the blood flows into the interstitial fluid, and then from there the fluid flows into these lymphatic vessels. And so the fluid is very similar. They're all what we call extracellular fluids, and they're very similar to each other in composition. Other than in the plasma, you have all of those blood cells where they don't get out into the interstitial fluid, and then in the lymph vessels, we tend to see just the lymphocytes. Okay. All right, so these lymph vessels, they don't make a circuit like we saw with the heart. We had arteries going away from the heart, and they led into the capillaries, and then the capillaries joined into veins, and they came back, and that's a circuit. But that's not the way it is with the lymphatic system. The lymphatic systems start in the periphery, and then they go back to the heart, and then they dump their fluid into the subclavian veins. So we'll take a look at all of that. But they all have an end point, and they come up, and they dump into the veins to get back into the blood. Okay. All right, there are lymphoid tissues that are dominated by these lymphocytes that we're going to see. We're going to talk about tonsils, so we'll look at a diagram with tonsils. That'll be on the lab exam. There's this other tissue that's called MALT, or mucus-associated lymphoid tissue. Those are where we have clusters of lymph nodes and lymph tissue in the digestive and the respiratory tract, the urinary tract, and the reproductive tract. So it's all of those lymphoid tissues are guarding like this really important area that has a lot of important organs. The lymph nodes we see, they're scattered throughout. We can see them all over the place. They are expansions of those lymphatic vessels. They tend to cluster in certain areas. So when we know the areas that they're clustering in, and again, they're acting like guards to that really important area where all the visceral organs are. Any disease that affects the lymph nodes is called lymphadenopathy. Lymphadenopathy. We have some other organs in here we're going to look at. We want to look at the spleen, and then we also want to look at the thymus. This picture here is showing the blood vessels as well as the lymphatic vessels. So you can see that blood is traveling down through the arteriole, and then it goes into the capillaries, and then that merges with the venules, and then it heads back to the heart. So it's a circuit. But the lymph vessels, they have an end point to them, and they're just collecting fluid. So the fluid moves from the plasma into the interstitial fluid and then into the lymph vessel. And then from the lymph vessel, that's going to head up back towards the heart and get dumped into the subclavian veins. This is just showing a picture of how the lymphatic vessels travel along with the arteries and veins. So again, anywhere, any tissue that you have arteries and veins in, you're going to have these lymphatic vessels. All right, this is showing some of the major lymphatic vessels that we have. And so we have a model that you'll have to identify these major lymphatic vessels. All of the lymphatic vessels are draining into these major lymphatic vessels, and then these two lymphatic vessels will drain into the subclavian veins. So let's take a look at what they are. So first of all, we have this one right here. You can see this green line going all the way up here, right? That is called the thoracic duct. And at the very end of the thoracic duct, we have this expansion, and that is called the cisterna chyli. So all the lymph vessels from the legs and the abdominal and pelvic area, they're all draining into this cisterna chyli, and then from there the lymph is going up into this thoracic duct, and then from there that thoracic duct is going to drain into that left subclavian vein, right? So if we look over here at this picture, we can see in this dark green area how much of the body that the thoracic duct actually drains. So it has the majority of the drainage. It's draining the fluid from the interstitial fluid and bringing it back to the heart and dumping it into the left subclavian vein. Then we have a smaller one over here on the right side, and we can just see right here, that is the right lymphatic duct, And so that right lymphatic duct has a smaller area, this light green area, the head, the right arm, and the right side of the trunk. That's what's draining, the fluid from there is draining into that right lymphatic duct. So when you look at our lymphatic model that we have, you're going to see these structures. You have to be able to identify the cisterna chyli, the thoracic duct, and the right lymphatic duct. This is a diagram that's going to be on the lab exam. This is showing the tonsils. So the tonsils are lymphoid tissue that are dominated by these lymphocytes that we're going to talk about. You have the one pharyngeal tonsil right here. Another name for the pharyngeal tonsil are the adenoids. So that's kind of right behind the nasal cavity. And then these other two are paired. So the palatine tonsil and the lingual tonsils, they're paired. There's one on the right and one on the left. So these palatine tonsils, those are the ones that you see at the back of your throat. Normally you don't see them, but then when they start to get infected, they swell. And sometimes they can swell so much that they start to touch each other. The right and the left side touch each other, and we say those are kissing. And then it makes it difficult to breathe. So at that point, then the doctor might decide to take those tonsils out. And then underneath, at the back of the tongue, we have these tonsils called the lingual tonsils. Again, there's a right and a left. And so this diagram will be on your lab exam. You'll have to identify those. This is what an actual lymph node looks like. And so the important thing here, I guess, is that the inside of the lymph node is dominated by lymphocytes. So there's both B cells and T cells in there. Those are the two types of lymphocytes. I'm not going to ask you in what layer are the majority of T cells or the majority of B cells, but they're packed in there pretty tightly. And if we look, we can see that there's some afferent vessels bringing the fluid in. So afferent means arriving, right? So, and there's several of them. So there's a lot of fluid from these lymphatic vessels, a lot of lymph, which is what the fluid is called, lymph. A lot of lymph is flowing into that lymph node. But then there's only one efferent vessel. So the efferent vessel is carrying the fluid out. And there's only one of those. So what does that mean for the flow of that lymph? It's going to slow down. It's like a highway merging. All these highways are merging together, and they're going to slow down, and the fluid is going to get caught inside the lymph node so that those lymphocytes can check to see if there's anything abnormal in there. And then we'll allow the fluid to leave and go into one of those major lymphatic ducts. When the lymph nodes are active, the T cells and the B cells are going to be proliferating like crazy. They're going to be dividing, and then that lymph node is going to get swollen. And so when we see that, this is a child here, and we can see how their lymph node is swollen. And so that's what you feel for in those areas where the lymph nodes tend to aggregate. This picture is just showing the thymus. So the thymus is a gland that sits right above the heart, and it's bigger in infancy and childhood, and then as a person gets older, it shrinks. As a person becomes an adult, it starts to shrink. So what's happening in the thymus is that's where T cells are maturing. All of the blood cells are actually produced in the red bone marrow, but then they mature in the thymus. The T cells mature in the thymus. And so there's a lot of actively dividing T cells in that thymus. So the thymus is bigger as a child and an infant because they're just experiencing new pathogens for the first time. And after a while, there's T cells that are produced that are directed towards those pathogens. So there will be fewer and fewer new pathogens that a person is introduced to as an adult. Another lymphatic organ is called the spleen. So the spleen sits right behind the stomach. The spleen is the largest collection of lymphoid tissue. It does a number of things. There are phagocytes in there, macrophages, that are going to remove abnormal cells. It's also the area where iron is recycled, iron from broken down red blood cells. It's going to be stored in the spleen. And it also contains T cells and B cells that will initiate the immune response. And we'll talk about the immune response in a little bit. The other thing is, though, the spleen has both what we call red pulp and white pulp. And the red pulp contains a lot of red blood cells. So there's a lot of red blood cells in the spleen. The white pulp contains lymphoid nodules or lymphoid tissue. But if the spleen erupts, then this red pulp is going to erupt and red blood cells are going to be leaking out like crazy. That's internal bleeding. And that will lead to eventual shock because the blood pressure will drop. So that's like an internal hemorrhage. This is just another look at the spleen. If they do remove the spleen, that's called a splenectomy. A person that has like mononucleosis, they are going to have to be very careful. Their spleen is going to be really swollen. And so any amount of contusion or any physical damage to that spleen could cause it to burst. It's very sensitive at that time, and then they would end up with internal bleeding. Okay, so now we're going to talk about immunity. Okay, immunity. So immunity is how your body is able to fight against any cells that are foreign or what we call pathogens that are that the body sees as dangerous to its homeostasis. And there's two different types of immunity. We have innate immunity. And innate immunity is what you're born with. These are things, all these things your body is born with. You don't have to develop them at all. It is non-specific, so it is to protect you against anything that's foreign, any foreign cell, any pathogen. And then we have another type, which is called adaptive. And adaptive we'll look at second. First, we're going to go over innate. And adaptive immunity is very specific. So there's cells that are the T cells and the B cells, and they're going to respond to specific pathogens, and we're going to see how that all happens. Usually, and when we're talking about T cells and B cells, the T cells are going to outnumber the B cells about 8-1. So there's a lot more T cells than there are B cells, but we will definitely get into that a little bit later. So let's talk first about this innate immunity. So you have basically 3 lines of defense, and the first line of defense is just not to even let a pathogen in, okay? So a pathogen, again, could be anything. It could be a bacteria. It could be a virus. It could be, a parasite. It could be toxins that are secreted from bacteria. I mean, these are all what pathogens are. And so the first thing your body wants to do is just to try to prevent them from even coming inside your body. So we have physical barriers. And those physical barriers are definitely your skin, so the epithelium, that's a barrier. It doesn't let anything come across it. There's also hair. Hair helps to prevent things from landing on your skin. And then we have some secretions. So we have oil from sebaceous glands. That will help to prevent bacteria or viruses landing on your skin. We have sweat glands that secrete a salty fluid, and bacteria can't live in, pathogens can't live in salt. And then we also have mucus membranes, because mucus membranes inside your mouth, inside your nose, inside your digestive, respiratory tract, reproductive tract, they help to produce mucus and prevent pathogens from getting in that way. So those are your physical barriers. So that's the first thing that happens. And then once something does get inside your body, then we have to go to a second line of defense, and that's going to be cells and cell processes, right? So now it's inside. Now there's cells in your body that are going to have to fight for you. So the first line, not the first line, this is in the second line of defense, but the first cells that become involved are the phagocytes. So phagocytes are any cells that can engulf anything that looks abnormal, right? They remove debris and they remove pathogens. If it looks abnormal, they're going to try to remove it, right? So we had the white blood cells. We have neutrophils, we have eosinophils, and we have monocytes. Those are the three white blood cells that are phagocytic, that can actually engulf things, right? And then the monocytes, when they squeeze out of the blood and they go into the tissues, then they're called macrophages. And because they're moving, they're called free macrophages. But then we also have some macrophages that kind of set up camp in certain tissues, and then those are called fixed macrophages. So we have like in the liver, we had the Kupffer cells. We now call them stellate cells. Those are an example of fixed macrophages. There's alveolar macrophages that just stay in the lungs around those alveoli. So there's some fixed ones that are just in specific tissues. Then we have the one of the T cells that's called a natural killer cell. So this is a T cell that is involved in innate immunity, all right? So innate immunity, again, means that they're just trying to stop anything that looks like a pathogen. It doesn't matter what it is, they're going to defend against it. So all the natural killer cells are going to try to defend against all the pathogens. And when the natural killer cell comes in contact with a cell that it thinks is foreign or it thinks that is dangerous, it has, we'll talk about antigens, but if it has antigens on it that it detects that are not of its own body, then they will shoot little chemicals out at that abnormal cell, and those little chemicals are called perforins. And once it shoots out, they're like little chemical bullets, and they pierce into the cell membrane, putting holes into the cell membrane of that abnormal cell, and pretty soon the abnormal cell lyses. So they're like, we say that the natural killer cells are part of immunological surveillance because they are, they're just out there surveying and looking around for anything that looks abnormal. Then we have interferons. And interferons, we're going to talk a little bit more in just a little bit, but interferons, they are going to resist viral infections, right? They're going to resist viral infections. They slow the spread of viral infections. That's what they do. All right, so they're going to be produced by any either lymphocytes or macrophages or any virus-infected cell. So what viruses do, and here's kind of a crude picture of what they do, but if this were a virus and it has a little bit of its own DNA or RNA, it's going to drill a hole into the cell. It's going to then, this hole that it's drilling, in the drill it's a hollow tube, and it deposits its DNA or RNA into the cell. Then that DNA goes into the nucleus and it gets incorporated into that cell's DNA and it takes over. And now it has the nucleus start producing viral parts. So now the cell is not taking care of itself. Now the cell is producing virus parts, and then it's assembling them together. And it keeps doing that until there are thousands and thousands of little virus pieces, and then it's going to make that cell explode. So when the cell gets infected by this virus, what the body cell is going to do is it's going to produce these little interferons. And these interferons then are going to go to other cells, neighboring cells, and try to slow down this process within those neighboring cells. They'll also try to slow down the process within this cell itself. So the thing with interferons is they're trying to slow viral replication. That's what they do. We have another, the next one is called the complement system. Again, this is still part of the innate immunity. They don't care what pathogen it is. But the complement system is made up of a series of these proteins. So you can see all of these proteins here. They're produced by your body, and there's a cascading event. There's different pathways. We're not going to learn any of that. You just need to know that these proteins that are produced by your cells are then going to attach themselves to a pathogen. So this red thing here is like a bacteria, and you can see how those little proteins are attaching themselves to the bacteria, right? And so a couple of things are going to happen then. First of all, those little proteins from the complement system act as a flag, and they're going to flag down a phagocyte. So a phagocyte might come by and, engulf it, right? And then get rid of it that way. Or it's going to weaken the membrane of the bacteria so much that the bacterial wall and the membrane will lyse, and then it's gone, right? So we can get rid of it that way. So that's what the complement system does. It attacks and breaks down the cell walls of bacteria. It attracts other phagocytes, and it stimulates inflammation. And that's the next thing we're going to see here is the inflammatory response. We're going to talk much more in depth about inflammation in just a couple of slides here. But the first thing that happens in the inflammatory response is whenever you have an injured or an infected area, inflammation is going to occur. And inflammation is a local effect. And it's going to try to do two things. It's going to try to prevent further injury to the tissue, and it wants to try to stop the spread of any bacteria. So there's a whole process that's involved. The mast cells are hugely involved with that. If you remember what mast cells are very similar to basophils. So basophils are your white blood cells. The basophils are in the blood, but mast cells are in the tissues, and they're fixed in the tissues, so they're not moving around, whereas the basophils are in the blood, and then they can move into the tissues. They both do the same thing. They're both going to release histamine and heparin. Okay, and then there's all of these things that happen with histamine and heparin. They're going to cause the blood vessels to dilate and the permeability of the blood vessels to increase. So we end up getting our blood flow increases. Phagocytes are going to be activated. Phagocytes are stimulated by histamine. And the permeability of the blood vessel increases. The complement system gets activated. We have a clotting reaction that's going to wall off the region so that bacteria can't get in, and bacteria, they just kind of stay clustered. Your temperature goes up, and then the specific defenses are activated, and that's the adaptive immunity, which we'll talk about later. But I am going to go further into inflammation. The signs of inflammation that we see are, do you guys remember what they are or know what they are? Whenever you have inflammation, cardinal signs. Yep, we have swelling. What else? Redness. What else? Heat and pain. Yep, so those are cardinal signs of inflammation. Redness, swelling, heat, and pain. All right, fever. Fever is a body temperature above normal. So normal is 37.2 degrees Celsius or 98.6 degrees Fahrenheit. Anything that's above 38 or above, and actually anything that's above 37.2 or 98.6 is considered a fever. Up to 100.4 degrees Fahrenheit, which is 38 degrees Celsius, we would say that's a low-grade fever. And anything that is above 39 degrees Celsius or 103 degrees Fahrenheit, that would be considered a high-grade fever. Okay? So fever is a good thing. It's raising the body temperature. These bacteria want to live at 37.2 degrees Celsius. That's why in our microbiology lab, the incubator that we have is set at 37.2 degrees Celsius. That's when they are able to replicate at their fastest rate. So they like that temperature. The higher temperature that your body gets, the less, the slower they're going to be at replicating. Okay? And then your body's going to be able to take over and kill them before they can replicate. So what causes fever? There are things that are called pyrogens that cause fever. Now some pyrogens can be what we call endogenous, and that means they're coming from inside your body. These are pyrogens that are produced in your body, and we're going to talk about these. Things like interleukin, interferon, so interferons, which I had already mentioned, are going to make you have a fever, and then tumor necrosis factor. And we're going to talk about these more in depth. And again, we call them endogenous because they're produced inside your body. But then there are other, what we call exogenous pyrogens, and those come from outside of your body. And so many of the bacteria, molds, yeasts, viruses, any of those things can induce fever. And so those are exogenous because they're coming from outside the body. There's one more cell that I want to talk about just briefly in the innate immune system, and it's called a dendritic cell. And so these cells, they hang out on the surface of your body. So they're on your skin, they're inside your nose, they're in your stomach, they're in your intestines. And so they are hanging out close to the surface that's exposed. Okay? And so they're phagocytic, so they eat pathogens. And they're very aggressive. And they carry, when they eat the pathogens, they carry that information. They're going to get back in the blood, and they're going to travel all the way to the spleen, and then, or the lymph nodes, and they're going to tell their intelligence. They're going to give their intel, their information to the spleen and to the lymph nodes so that they can start an immune response. Okay, so they pass intelligence. To the spleen and lymph nodes. Because inside the spleen and inside the lymph nodes are those T cells and those B cells. So they're going to give the information, say, look, this bacteria or this virus, this pathogen just came into the body. I wanted you guys to be the first to know. They are very lethal, and they're very intelligent. I want to talk about the differences between infection and inflammation, and then we're just going to move on from there. An infection is when your body's being invaded by microorganisms, and then they just keep multiplying. And so once they do that, we're talking about organisms like bacteria, viruses, parasites that are not normally found, are not normally present in your body, or they become in culture in higher numbers than what's usual. An infection can cause no symptoms, it could be, and then it's subclinical, or if it does cause symptoms, then it could be more clinically apparent and you would see clinical symptoms, okay? An inflammation, on the other hand, and this is what I'm going to be talking about here for a couple of minutes, it's, first of all, it's a local response to injury to the cells, right? So you end up getting capillary dilation, you get white blood cells entering the area, and then you get those 4 cardinal signs: redness, heat, pain, and swelling. It's designed, that inflammatory process is designed to protect the body from injury and invasion of pathogens. All right, so let's take a look at what happens with inflammation. So the tissue is injured, right? And the mast cells are activated. And the mast cells, we said, are a lot like basophils. They're going to release histamine and heparin. Histamine and heparin cause vasodilation and increased permeability. So cells can leave the blood and move into the tissues. So the first thing, let's look over here. These are the 4 cardinal signs: redness, swelling, heat, and pain. And then also clot formation will occur. But in addition to that, the mast cells are going to attract phagocytes. So the histamine that's released is going to attract phagocytes, especially the neutrophils. And if you remember, those neutrophils are the ones that are the first ones there at the site of an infection. They are going to remove debris. And then other white blood cells, phagocytic cells, are going to come and help to remove debris. The fibroblasts are going to start to repair the area by building new fibers and scar tissue. And then the other thing that happens is that cytokines are released. So cytokines they're in your textbook, in that chapter, but there's not a lot about them. We're going to talk about more of them later, but there are some cytokines that I want you to be aware of that you're going to need to know for the health field. So we're going to look at this one particular cytokine that's called a bradykinin. Bradykinin, when the tissues are damaged, bradykinin is released from the damaged tissues, and they are going to release arachidonic acid. Then arachidonic acid is going to convert to these things that we call ecosonoids. Ecosonoids are lipid hormones. They're local lipid hormones. You could read more about those in chapter 18, right, if you want to. These are the 3 main ecosonoids that there are. There's prostaglandins, thromboxanes, and leukotrienes. Prostaglandins, they are powerful vasodilators. They're going to dilate those blood vessels and bring more blood to the area. They cause inflammation, pain, and fever. Okay? They're also going to prevent needless blood clotting. Another type of ecosonoid is called a thromboxane. Thromboxanes, like the name implies, thrombocyte, they're produced by platelets, and they are powerful vasoconstrictors. They promote platelet aggregation, so they're going to promote blood clot formation. And then the third type of ecosonoid is a leukotriene. Leukotriene, like the name implies, they're produced by leukocytes. And so whenever prostaglandins and histamine are produced, these leukotrienes are going to be produced as well. Leukotrienes you're going to hear about in the health field because they cause bronchial constriction. And so with people that have asthma or rhinitis, they usually have an overproduction of these leukotrienes. And they're also going to attract neutrophils. So these are these are ecosonoids. And so the big deal here is the reason why I'm having you understand this and learn this is that when arachidonic acid, when you have an injured tissue and arachidonic acid is released, arachidonic acid is going to be converted to ecosonoids. And they're converted that way. As soon as they're converted to ecosonoids, you get inflammation. So that's the big story, is that you're getting inflammation with these ecosonoids, right? But that conversion requires these enzymes that are called Cox enzymes. They're Cox enzymes. And so there's two different types of Cox enzymes. COX-1 is this is an enzyme that's always active, so it's always being released. It's found in the kidney, the stomach, and the platelets. It's used to make thromboxane. Okay? But the big thing here is that COX-1 enzymes protect the stomach. They increase mucus production and they decrease gastrin. So they're helping to protect that stomach lining from acid. But the COX-1 enzymes are constantly working, always converting arachidonic acid to those thromboxanes. It's just always working. The COX-2 enzymes, these are the ones that are going to be, used to make prostaglandins, and they're going to be increased whenever the tissue has trauma, ischemia, or pathogens are there. So we get an increased amount of COX-2 enzymes. They're found in macrophages, leukocytes, fibroblasts, all the cells that we would normally see in a tissue that's inflamed. Okay? So when you go to pharmacology and you start looking at COX inhibitors are, they are going to inhibit the COX enzymes. So they're going to stop arachidonic acid from converting to eicosanoids. So they're going to stop the inflammation. They're not going to allow arachidonic acid to become eicosanoids, right? So some of the COX inhibitors that are out there, like aspirin and ibuprofen, or like Motrin, Advil is ibuprofen, they block both COX-1 and COX-2 in the enzymes. They block them both. So they're blocking COX-1. And in COX-1, what I said was that COX-1 is there to help protect the stomach, to increase mucus production and decrease gastrin. So if we're blocking that and we're giving a person aspirin or ibuprofen, they have a higher chance of stomach problems, ulcers, stomach upset from taking these. There's other NSAIDs out there, non-steroidal anti-inflammatories, like meloxicam, and they're specific and will only block COX-2 enzymes. So now they're just blocking COX-2 enzymes, the ones that are increased with trauma, ischemia, and pathogens, but they're not blocking COX-1 enzymes, so you still have the protection of the stomach. So then that's the end of innate immunity, and now what I want to do is I want to look at adaptive immunity. All right, so adaptive immunity is going to deal with T cells and B cells. And there's different types of those cells. This is just kind of giving an overview of what happens with the T cells and the B cells. The T cells and B cells are involved in what we call specific defenses. They're going to respond to a very specific pathogen. So one T cell might be responding to the Staphylococcus aureus bacteria. Another one might be responding to a varicella virus, right? So they're responding very specifically to things. We're going to look at the whole pathway on how these things are actually responding. The T cells, we say, are cell-mediated immunity. They're involved in cell-mediated immunity because they can't really recognize bacteria and viruses that are just out in the interstitial fluid. They can only recognize those pathogens if they've infected the body first, right? They have to infect the body first. And then the B cells, we say, are part of antibody-mediated immunity because once those B cells become activated, they are going to become plasma cells, and then plasma cells are going to produce antibodies. So that's why we say T cells are cell-mediated and B cells are antibody-mediated. Quick talk really fast about antigens. So any antigen we have antigens that are found on top of like bacteria. So that's a little bacteria, and here are the little blue things. These are three-dimensional proteins that are found on the cell surface. And you have antigens on all of your cells in your body too, but your immune system is looking at those antigens and saying, well, those antigens are normal. They're part of your body, so I'm not going to worry about it. But when a bacteria invades your body, then your immune system, those T cells and B cells, are going to look at that cell and say, you know, those antigens look very different. They're not part of us, and they're going to mount an immune response, right? So antigens can be either the entire pathogen, or it could be parts of the pathogens, or it could be like toxins that the pathogen is secreting. And that's what your immune cells are going to respond to. There's different types of immunity. We have either active immunity or passive immunity. Okay. Active immunity means that you come in contact with an antigen and your body responds. I'm going to show you how it responds in just a little bit, but it responds. The T cells are going to become activated, and the B cells are going to become activated, and they're going to mount this huge response, this immune response against that antigen. That's active because your body is actually actively doing something to stop it. Passive immunity, on the other hand, this is just the transfer of antibodies. So a person is receiving antibodies from somewhere else, from another person, or from, we find antibodies in some serum from other places, okay, if it's a health professional giving the antibodies. So active, in other words, that person is actually having to produce the antibodies themselves. Passive means that they're just being given the antibodies, right? These are different types of immunity. Naturally acquired means it's just coming through the environment. Active means it's the antigen that's coming through the environment, right? So naturally acquired, active. So how do you think this immunity would be transferred? How do you think a person would develop naturally acquired active immunity? Yeah, they're just coming in contact with some antigen. So they're shaking hands with someone, then wiping their hands on their nose, or they're wiping their eyes. They're somehow someone's sneezing on them, and it's coming across the environment, and so they're getting the antigen, right? And then their body has to build those antibodies to try to fight that antigen, okay? So that's naturally acquired active immunity. Then we have artificially acquired active immunity. And this is where this artificially acquired means that a health professional is the one to give the antigens. So when does a health professional give antigens? That's right, that's vaccines. So that's when a vaccine is either a live virus, which normally they haven't been doing that. They used to do the polio virus live, or it's attenuated, in which means that the virus has been killed and broken apart, and then the antigens are just transferred to the person. But then they're able to develop an immunity against that antigen. And then we have naturally acquired passive. So naturally acquired passive, naturally again means it's coming through the environment. Passive means that it's antibodies that are being passed. When do we see antibodies being passed through the environment? Breastfeeding, or even through the placenta, because antibodies are small enough to cross that placental lining. And then the last one that we have is artificially acquired passive. So artificially acquired passive, again, this is through a health professional, but it's passive, so it's antibodies that are being given. When would a health professional give antibodies, like immunoglobulins? Snake bite when they, right, so it's kind of like an anti-serum where they have to, if there's a snake bite and the person doesn't have enough time to build up their own immune response or they'll die, you would want to give them antibodies, or in the case of rabies. Person doesn't have enough time to build up that response, which can take days, right? And they don't have that time. So then a health professional would just give them these immunoglobulins. This adaptive immunity, there's 4 properties that we're going to see here. The first one is that adaptive immunity has what we call specificity. Specificity just means that the cells have specific targets. It's not like that natural killer cell that will just target anything that looks abnormal. Each T cell has specific targets that it's looking for. Each B cell has specific targets that its antibodies are going to look for. The second thing, though, is that this adaptive immunity has versatility. Versatility means that it's okay that it's that specific because there are several different groups of these cells that can respond to many, many antigens, right? So it's very versatile, even though it is so specific. It can still handle all those antigens. The third property is memory. So adaptive immunity has these memory cells, and these memory cells can wait and not do anything this infection, but the next time you get infected by the same thing, it can mount an immune response much faster than it did the first time. So it'll remain inactive until the next invasion. And then finally, we have tolerance. And tolerance just means that your immune system isn't going to attack your own cells. Your cells have antigens that we call self-antigens, and they won't attack your cells. They know the difference between your cells and foreign cells that enter into your body. Let's look at T cells to begin with, all right? Because T cells are going to become activated first. And so T cells, there's several different types of T cells that we're going to see. I'm going to mention them as I go along. But the T cells have to be activated before they can even do anything. So in order for them to be activated, they first have to have another cell present the antigen to them, because the T cells can't respond to anything in the interstitial fluid. They have to have another cell present the antigen, and we call that antigen presentation. So we're going to just look briefly at that. Then the T cell has to recognize. So first another cell has to present the antigen, and then the T cell has to recognize that antigen, and we call that antigen recognition. And then finally, once that T cell recognizes that antigen, it's then going to become activated, and at that point, it will start creating clones. All right, so let's just look at these. I'm going to give you the whole story, and then I'm going to go back and I'm going to break it down a little bit. All right, these are cells in your body that can present the antigens. Right? So this is just a nucleated cell right here. This is a macrophage. Here's another lymphocyte. So all of these cells have the ability to present the antigen. And they present the antigen using what we call Mhc proteins. So Mhc proteins are proteins that are produced inside the cell and then are brought out to the surface of the cell, and then the antigen is presented on that Mhc protein. So that's all that Mhc protein is. There's a class 1 and a class 2, which we'll talk about in just a minute, right? So the first thing you need is one of your cells has to be infected in order for the T cell to respond. They get infected, and then they present that antigen, just like Simba, right? So they're presenting it. Now the T cells, they can recognize those antigens. And once the T cells recognize the antigens on those other cells, they become activated. Okay? Now, when those T cells become activated, they quickly start cloning, and their clones differentiate into the different types of T cells. These are the different types of T cells. Cytotoxic T cells. Cytotoxic T cells, they directly kill other cells. Right? Memory T cells. These are memory cytotoxic T cells. They just stay waiting, they stay inactive. They're not going to do anything until the next infection, but the next time they are infected with that same antigen, they will start dividing and cloning, and they will be able to attack those cells that are infected right away. The regulatory T cells, these are regulatory cytotoxic T cells. These T cells are going to slow down the immune response. They do that because they realize that at times an infection might not be short-lived, it might be a longer ongoing infection. So they're going to want to slow down the immune response so that you can keep fighting long-term. The T cells can also differentiate into helper T cells. Helper T cells are, what they do is, there's several different things that they do, but one of the things they do is they are going to activate B cells. When B cells become activated, they produce plasma cells. Plasma cells produce antibodies, and there's 5 different types of antibodies. So we're going to work our way through this whole process so that you can see it a little bit slower and see what's going on. We're going to start with looking at the T cell here. There's 2 different classes of those Mhc proteins. There's a class 1 and there's a class 2. I'm going to discuss class 1 here, and then on another page I'll discuss class 2. Okay? So class 1 Mhc proteins are found in all the nucleated cells in your body. In every cell in your body that has a nucleus, those cells are able to make these Mhc proteins. And Mhc proteins are those proteins that help to present things on the surface of the cell. Right? So every day your cells have debris and have things that are not so toxic in them. And so the cell produces these Mhc proteins, and then those proteins are going to display that debris outside of the cell. And a T cell that sees that will say, well, that just looks normal. That's just normal debris, and it will ignore it. So that's constantly being done, and that is an Mhc class 1 protein because it's in all your nucleated cells. All right? But sometimes your cells get infected, and then the Mhc class 1 proteins are still involved. So this is one of your body cells here. And here's the nucleus is making these Mhc class 1 proteins. They're being packaged up by the endoplasmic reticulum. And here we see a virus. The virus has infected the cell. The nucleus is creating new viruses. Your lysosomes are going to tear those viruses down into little antigens, and now that Mhc1 protein can present it. So the Mhc1 is presenting now an antigen. It's not just presenting debris, it's presenting an antigen. So this T cell now recognizes that antigen, and the T cell is going to become activated. Okay. when a T cell becomes activated by an MHC1 protein, it becomes a CD8 T cell. It's going to start dividing and cloning, and its clones are going to be called CD8 cells, CD8 T cells. And the CD8 T cells can be either active T cells, active cytotoxic T cells, memory cytotoxic T cells, or regulatory cytotoxic T cells. So you're linking some things together here. So to remember this, you just have to remember the MHC class 1 protein will activate a T cell that will produce the CD8 cells. The CD8 cells are those cytotoxic T cells. So what do the cytotoxic T cells do then? They're going to attack this infected cell, and then they're going to go off and attack other infected cells that are infected by the same virus. All right, let's look at the Mhc2 protein. So this is MHC class 2. So these proteins, these class 2 proteins, they are produced by what we call antigen-presenting cells. So these aren't just your normal cells in your body that have been infected. These are cells like macrophages or lymphocytes. Here's an example here of a macrophage. So macrophages engulf things. So this cell was, it tried to become infected. It didn't just happen to get infected. It tried to get infected. It engulfed the bacteria. That's what the macrophage does. It engulfs it. The bacteria then, that vesicle joins with a lysosome and then gets broken down into little antigenic parts, right? The nucleus produces the MHC class 2 protein. The endoplasmic reticulum packages it up into a vesicle. And then the MHC class 2 protein will present that antigen on the surface of the macrophage. So here we see it presenting it on the surface of the macrophage. Now here's a T cell that recognizes that and will become activated. And so when a T cell becomes activated by the antigen, by an antigen-presenting cell, it will start cloning, just like we saw with the MHC class 1. But now the clones are CD4 T cells. CD4 T cells. And the CD4 T cells are helper T cells. They're helper T cells. So they're not going to directly kill any cells. Helper T cells don't directly kill. The only ones that directly kill are the cytotoxic T cells, and those are CD8 T cells that were activated by MHC class 1. These are helper T cells that were activated by the MHC class 2. Okay? So this is what helper T cells do. Helper T cells, they are going to do these things. First of all, let's start over here. They are going to attract the cytotoxic T cells to the area. So even though they can't directly kill, they will attract the cytotoxic T cells that are already activated to that pathogen so that the cytotoxic T cells will come to the area. Okay? They will also attract other macrophages to come to the area so that they can engulf more of that pathogen. And they also activate B cells. So the next thing we're going to look at is B cell activation. Before the B cells can become activated, they have to be sensitized. Right? So let's look at what happens with the B cells. So here we have a B cell. The B cell looks very similar under a microscope to the T cell because they're both lymphocytes. And there are millions of B cell populations in your body. And each of the B cells have these little antibodies, these little Y-shaped proteins sticking off of their or out of their cell membrane. Each B cell has thousands of those antibody molecules. But the B cell, before it can become activated, it has to be sensitized. So a pathogen, like a bacteria here, a bacteria binds to that antibody on that B cell. And then the B cell displays it on the cell membrane with another Mhc protein that we're not going to name. So now the B cell is displaying that antigen. And when that activated helper T cell comes along, it will be able to activate that B cell. So, short story, the B cell becomes sensitized by the pathogen, and then it becomes activated by the helper T cell. 2 steps. B cell becomes sensitized by the pathogen / antigen. And then 2, the B cell becomes activated by the CD4 helper T cell. Step 3 then, that activated B cell starts to clone and divide and divide and divide. And as it's dividing, its daughter cells differentiate into plasma cells. So step 3, the B cell produces plasma cells as it divides. Step 4, the plasma cell produces antibodies. So those are the steps to the B cell activation, and the end result is we end up with all of these antibodies. Okay, so there's 5 different types of antibodies, and we're going to go over what those 5 types are. I like to use the acronym MAG. IgM, IgA, IgD, IgG, and IgE. MAG. Okay, so here we have IgM and IgA. IgM is pretty large. You can see that it's actually made up of 5 different antibodies. Antibodies are these Y-shaped proteins. So there's 5 there. They're the first class of antibodies secreted after an antigen is encountered. So your IgM, they increase in numbers quite rapidly. They accelerate production quite rapidly, and then it declines quite rapidly as well. Okay. The anti-A and anti-B antibodies in blood, those are IgM molecules. Those are IgM antibodies. Okay, those are examples. Then we have IgA. IgA is found in glandular secretions. So you find those in mucus, in tears, saliva, semen. IgG. IgG is the largest and most diverse of the antibodies, and it accounts for about 80% of the antibodies. So they're responsible for resistance against many, many pathogens. They can cross the placenta. They can cross the placenta. So the mom can give protection to the baby. This is the anti-Rh antibodies. They are the IgG antibodies. Then we have IgE. IgE is it is going to be increased stimulated when you have an allergic reaction. All right, so IgE, you want to remember that you see those with allergic responses, and then we have IgD. And IgD is the ones that are found on those B cells. So when I was drawing the B cells and I showed you all of those little antibodies sticking off of it, those are IgD antibodies. Okay. So the last thing I want to go over are these things that are called cytokines. Cytokines. Cytokines are released by lymphocytes and macrophages. Okay. One of the cytokines I already talked about, I said it was perforin. So perforins are these little chemicals that the cytotoxic T cells, they shoot out in order to kill an infected cell. But the ones I want to talk to you about right now are other cytokines, and these cytokines, most of them are produced by lymphocytes and macrophages. Some of them can also be produced by endothelial cells, fibroblasts, or astrocytes. But let's go through each one of them. You're going to end up hearing about these at some point. Lymphocytes and macrophages, they release interleukins. So interleukins are a type of cytokine. Interleukins do a number of things. They increase the sensitivity of the T cells to the antigens being presented by the macrophages. They just make them more sensitive. They make T cells more sensitive. They activate the B cells to produce plasma cells. Okay. They speed up the activation of B cells to produce plasma cells. They enhance your innate immunity. They cause inflammation. They cause scar tissue to start forming. They cause your body to get a fever. They cause mast cells to start being formed. Right? So they're enhancing that innate immunity. Right. Some of them are going to shorten the immune response, some don't. So the big things for interleukins is they increase the sensitivity of T cells, they are going to speed up the activation of B cells to produce plasma cells, and they're going to enhance innate immunity. Interferons we already talked about. These are also cytokines. They're released by virally infected cells. They attract natural killer cells. They want those cells to be killed, those infected cells to be killed. They slow inflammation. They stimulate macrophages. Interferons you're going to hear about in the health field. Interferons can be used to treat some types of cancers. They give interferons to patients to treat malignant melanoma, to treat bladder cancer, ovarian cancer, or even leukemia. Interferons are also given to AIDS patients who have Kaposi sarcoma, which is another type of cancer. Right? So you'll end up at some point hearing about interferons. The last type of cytokine that I want you to know about is the tumor necrosis factor. The macrophages are going to release this tumor necrosis factor. Cytotoxic T cells can also release it. So you got to think of these cytokines as like little bullets. And what are the little bullets doing? These cells are shooting out these little bullets at other cells, and what are they trying to do? So the tumor necrosis factor, like the name implies, it slows tumor growth and it kills sensitive tumors. It also stimulates white blood cell production, those granular ones, BEN, basophils, eosinophils, neutrophils. And they promote that eosinophilic activity. Tumor necrosis factors act as pyrogens, so a person who's having these TNFs released will end up with a fever. They also increase the T cell sensitivity to the interleukins.